Well, good afternoon, everybody, and thank you so much for being here still on this beautiful, sunny Saturday afternoon. This is actually the first time I saw sunshine in Berlin for this week. So I really appreciate you guys stay in here, inside still, while you can actually go outside and enjoy the sunshine. So I am with Applied StemCell, and prior to Applied StemCell, I did spend many years at Stanford University. So I was at Stanford for 19 years, and then I joined this company a while ago. And so today I will not talk about stem cells, but I will talk about technologies to target gene insertion and gene editing for application in gene therapy, and also include potentially for anti-aging therapies. So this cartoon shows just a layman's view of what gene therapy is. So gene therapy is really an old idea, but just recent years it entered into a new era which provides a curative medicine for therapies. So this cartoon says scientists are hoping to isolate a big hand gene so that people can fly. So that's just a concept, but that's actually really kind of reflect what gene therapy is. So what exactly is gene therapy? So gene therapy is addition, alteration, or removal of genes within cells and biological tissues to treat disease. So basically, a person who has a defective gene, so which causes a specific disease. And then the gene therapy method is basically introduce a therapeutic gene or a healthy gene into the cell, therefore the cell can function again. So the concept is pretty simple. However, the procedures leading to the therapy is pretty complex. So there are two types of gene therapy methods. So they're mainly in vivo and ex vivo. So ex vivo is more of an indirect gene therapy, and it usually uses targeted cells as a carrier for carrying therapeutic genes or modified genes. And then these cells are infused back into a patient. So that's how the ex vivo gene therapy works. And then in vivo therapy is a more direct gene therapy method. So in this case, so a DNA is delivered directly into a human body to specific tissues, either by viral vector or non-viral vector to specific tissues where the gene indication is manifested. So just a bit more about ex vivo gene therapy, and sometimes it's also called cell therapy because the carrier of the gene is a cell. So basically, this describes a procedure of what a cell-based gene therapy is. So cells are taken out of a patient or from a healthy donor. And these cells are, I don't think this pointer works, but anyway. And these cells are then manipulated ex vivo in a petri dish. And in some cases, these are modified by lentiviral vector transduction, which carries a gene into the cells. And then these modified cells are infused back into the patient. So this is how the cell therapy works. And if the cells, the donor cells, are from the patient themselves, it is called autologous transplantation, so autologous cell therapy. And if the cells are taken from a donor, a healthy donor, and this is an allogeneic cell therapy. So you may be more familiar with the CAR-T immunotherapy. So CAR-T immunotherapy is probably the most advanced cell therapy nowadays. So basically, for CAR-T immunotherapy, so T cells are isolated from a patient, a cancer patient. And these cells are manipulated by integrating a CAR, which is basically an antigen, into the cells. And now these cells can recognize and attack cancer cells. And these cells are then replicated into millions of these CAR T cells in the lab, and then infused back into the cancer patient. So that's how our CAR T immune therapy works. You probably heard about it a lot. So now for in vivo gene therapy, because we don't have cells to carry the gene into a human body, so it's a bit more difficult to do, but there are ways of delivering genes in vivo directly to specific tissues. So the most common delivery system is a viral vector. And the most among the viral vectors, the most common one is adeno-associated virus, which is AAV. And nowadays, people are also developing many different forms of nanoparticles to carry DNA into a cell inside the body. And so sometimes naked DNA is also used to treat patients directly. So I think one of the earlier talks by Doris, she was using DNA vaccine to treat Alzheimer's disease. So that is by direct nucleic acid delivery, and her system was a pump system to deliver DNA. And so these different delivery systems will have specific tissue tropism so that it can deliver specific genes to these tissues, including the brain, muscle, eyes, liver, and the bone marrow. So now a very brief history of gene therapy. The reason I want to do that is that basically gene therapy is a very old concept. So it initially started in 1960, but it really didn't progress until the last few years. Okay, so like I said, the concept was first introduced in 1960, and it didn't really get into the human until 1990. So that was the first success story with gene therapy. That was to treat sickle cell anemia. So that was a success story. However, 9 years later, there was a very sad case in which gene therapy actually caused a death. And this really set back gene therapy in the United States, at least. So I just quoted here my Cornell alumni, Carl Sagan, saying, "You have to know the past to understand the present," and I added the future. So now, just look at in a very short period of time. So the first gene therapy drug that was approved was in Europe, and it was called Glybera in 2013, and it was developed by a Dutch company called Unicure. And so this drug was priced at $1 million. So gene therapy so far is the most expensive drug in the world. So this basically got a name, the highest, the most expensive drug in history. However, it really provides a proof of concept how gene therapy can work for certain diseases, especially genetic diseases. So this is basically a summary of history of 53 years. So we only saw one drug that was approved. That was in Europe. So what I want to show you is that just in 1 year, we had 3 drugs, gene therapy drugs, that got approved in the US. So you can see how fast this field has evolved. And it's going to continue evolve even faster. Okay, so this is really a revolutionary therapy that's coming into the space. So in 2017, FDA, the US FDA approved 3 cell and gene therapy products. So 2 of them are CAR T therapy, which are for cancer treatment. So Novartis and Kite, they both developed CAR T therapy, and Kite was later acquired by Gilead. And these are 2 CAR-T therapy for leukemia or lymphoma. And then the third drug is a gene therapy drug called Luxturna. So this is a gene therapy for blindness, a very rare disease. So I put the pricing tag here. So you can see the price tag is still pretty high, even though it's lower than $1 million. But the pricing tag is still pretty high because just the development of these drugs is very costly. So, but however, if you look at it, because gene therapy, the idea of gene therapy is really one-time treatment, a lifelong cure, right? So if you have to use a drug on a weekly basis, for example, you add up the cost for a lifetime. That's a lot more than a one-time treatment here at $500,000, for example, right? So it's still a cost-effective way of medicine. So, so Spark, which, like I said, is the first company that did in vivo. These are ex vivo gene therapy. Spark did it in vivo, directly delivered into the eye. And this company was just acquired by Roche about a month ago at $4.7 billion, $4.7 billion, which is more than 2 times of their stock pricing. So you can see the valuation of a gene therapy company is very high, and so it's very accepted now by large pharma. So here I want to give you a taste of how many more gene therapy products now in the pipeline. And so for various diseases, including blood disease and metabolic disease and muscular dystrophy, many different indications, and eye disease. And the list is very long, so I just want to give you a taste of it. And the bottom line is that this is really coming of age for gene therapy. It's the time. And the promise of gene and cell therapy is really tremendous. And to meet the demand of all these products that are in the pipeline, the US FDA has added 53 more people to just review these product IND filing and clinical trials. I can give you one example. So 2 years ago, my company, we applied for pre-IND, and we had 10 people from FDA to the meeting, to sit on our pre-IND meeting. And just 1 month ago, we applied for another pre-IND. We have 20. We have 20 people from FDA to our meeting to review our product. So the support from FDA is really tremendous, and they really speed up the process of reviewing, just because there's such a long list in the pipeline that need to their review. So now I said all these wonderful things about gene therapy. So now I'm on the other side of that, which is there are limitations of gene therapy, right? So even though it's a one-time delivery of genes, however, it's still short-lived. I say short-lived. It's all relative, right? Long-term, short-term. So we don't know how these genes are going to stay active and expressed in human body. Right? We don't have enough data to see that. The longest data right now in clinical trial is about 3 years. We see effects for 3 years, right? So, but if you think about for pediatric patients, that's not good enough. You cannot treat pediatric patients. You need to show much longer effects, right? So that's still a big challenge for gene therapy. And then also the genes, they're episomal, or even if they're integrated, and they integrate in a random fashion, you don't know which part of the genome the therapeutic gene goes in, so it may not express. If the gene is not expressed, it's not effective. It's not going to work, right? And so because this is random gene insertion. And then at this moment, it will be very challenging to treat diseases that are caused by multiple genes. And so far, we choose indications that's only caused by single gene, right? So you deliver one gene, then you cure the disease. If it's caused by very complex multiple genes, such as aging, right? And it may take us many years to reach that point. So there are also immune response, because you put in a foreign gene into a human body, the natural reaction of a human body is to reject those new proteins. So these are the clinical challenges that we're facing in doing gene therapy. So now I want to switch, just move to the a little bit advanced technology, which is gene editing. So just for those of you, I don't want to confuse it. So gene therapy is put a gene, introducing a gene. Gene editing is really not just introducing the gene into a cell, but also this gene is integrated into your genome. So that means when cells divide, you don't lose your gene. Okay, so it's basically it becomes part of your chromosome, and keeps dividing, and you still have that gene. So gene editing and gene therapy difference is gene editing, actually the gene gets integrated into your chromosome. Okay, so this was a big breakthrough, especially when CRISPR-Cas9. You probably all heard about CRISPR because this is almost like everywhere. So CRISPR came out in 2013. And this field just exploded, just because the power of CRISPR, which acts as like a genetic scissor. So it cuts the DNA and it repairs the DNA very precisely in a site-specific manner. So, okay, so this is the only slide actually I asked you not to take a picture. So I tell you why. Because, I'm just using an X-ray photo as analogy. So this is a broken collarbone X-ray. That's my daughter's X-ray. So that's why I told you not to take picture, because I didn't get her permission to show her X-ray. Okay, so she broke her collarbone while skating, right? So, and you see the collarbone broken into 2 pieces. And so it cannot be healed by itself. So the surgeon, basically connected 2 broken collarbone by using plates and nails. Now it becomes one again. So you can imagine that's what happens at the molecular level inside a cell. Okay, so inside a cell, that's how CRISPR works. Okay, so basically CRISPR uses an enzyme to break the DNA, then at the same time insert a foreign DNA, a therapeutic gene, into this locus, and then heals it. Okay, so that's how it works. So CRISPR is like a surgery, a microsurgery also, right? All right, so besides CRISPR, there were actually other technologies which were not as popular because they were hard to use, and you really need a special skill in order to do it. So those include zinc finger nucleus and talons, and they are in the same category. They all work similar in terms of how the enzyme works. And then, so what I really want to introduce is our technology, which is based on integrase, and we trademarked the technology Targa. And this technology is a site-specific gene insertion technology, so complementary to CRISPR. And so this technology is unique to my company, Applied StemCell. So I will talk a little bit more about how integrase works in the several slides later. So again, I kind of described to you already how CRISPR works. So it has 2 components. So one is an enzyme, which is a nucleus. So the function of that nucleus is to make DNA breaks. So it breaks up the DNA. So the cells have to repair the DNA, right? So during repair, if you put a foreign DNA, it will go in through either homologous recombination or just end joining, just join the DNA to repair the DNA. And the other component of CRISPR is called a guide RNA. So the function of guide RNA is just really to do site-specific targeting. So you don't want to just have CRISPR going to Cas9 going to your cells, cut up your DNA, right? So the only way that the enzyme will work is that the guide RNA has to bind a specific site in your genome and then make the cut. So it's a site-specific cut. It's more targeted cut. All right, so there are a bunch of companies established because of CRISPR technology. And so FDA has recently accepted 2 IND for clinical trials. One is CRISPR therapeutics. So they're using CRISPR technology to treat beta thalassemia and sickle cell anemia. And the other company is Editas. So they're using the CRISPR technology to treat blindness directed in the eye. So for those lawyers in audience, because I met a couple lawyers, so the interesting part of CRISPR is that there's an ongoing patent fight between the major IP holders. So one is MIT Broad Institute, the other is UC Berkeley. And these two companies basically still have big patent debate and infringement debate. So that's going to keep going on for many years. So we're still not clear who owns what in what country, so in terms of the IP landscape. So CRISPR Therapeutics was founded through the UC Berkeley route, and the Editas was founded through the MIT route. So you can see, and then these are the major ones, but there's so many other ones, basically, now public companies because of CRISPR. And just looking at CRISPR Therapeutics' valuation right now is $2 billion. They don't even have a product. So you can see the promise of the gene editing and gene therapy. All right, so I want to use one example, which is aging-related, that uses CRISPR technology. So this work was done by Juan Carlos Belmonte, who is a PI at Salk Institute in San Diego. And so his lab basically used CRISPR technology they show they can extend lifespan of mouse by 25%. So that's pretty incredible for mouse. So they have a mouse model that has progeria syndrome. And so what they did was they just put CRISPR reagents to treat those animals. So if you look at those, this is looking at survival. So if you look at the red line, so that is treated with CRISPR-Cas9 and guide RNA, you see a long extension compared to the untreated one, which is the blue-green line here. So this is a pretty incredible result. They were able to extend the mouse life by from 20 weeks to 25 weeks. So this is just one example on how to using CRISPR to potentially to treat. This is actually a disease. Those are the people who have genetic disease that has a mutation in lamin A, and they have accelerated aging. They don't live beyond 15 years old, so which is very sad. So this is actually a disease, and then can be treated, potentially translated into human. So now I want to just come back to introduce to you about what we do at Applied Stem Cell Therapeutics. So we're really a technology company and genome editing company, and we use that technology both for research and therapeutic development. And even though at the current moment, the therapeutic development may not be directly related to aging, but what I want to say is the platform that we're trying to set up is totally applicable for anti-aging therapy in the future when the time comes. So this is our pipeline. Like I said, so for right now, we're focusing on hemophilia and GVHD. So these are the programs that are closer to clinical stage. And then we also have some early discovery programs. So I already introduced this slide about how gene therapy works. So this is our technology platform. So we have an improved CRISPR technology called the CRISPR-Alt NHEJ. We filed patent back in 2016 for gene knock-in. And we also have delivery system, which is novel AAV capsids, which we licensed from Mark Kay's lab at Stanford. And we have our own integrase technology. We're developing variants using a directed evolution technology to find better integrase enzymes that can be used for human therapy. So just a little bit about target integrase. So target integrase, basic integrase catalyzes the two major features, irreversible and site-specific gene insertion. So this is just schematic. Basically, this integrase recognizes two sites, attachment sites, ATDP and ATDB, and the recombination between these two sites results in the integration of the gene of interest here, which is in black. And at the same time, it creates two new sites, two hybrid sites. So these sites can no longer be recognized by integrase. So the direction is basically only one direction, only functions as integrating the DNA. The reverse reaction does not happen. And another main feature is that we're able to insert large transgene up to 20 kb. So I want to emphasize this because CRISPR, although it could insert DNA, but at very small pieces, okay, very inefficiently. So then Target came in for large piece insertion. I see you're here, so I have to speak faster. Okay, so I'm going to skip these. So this is just therapeutic application using our Target technology. And now I want to jump into this enzyme. So BXB1 is another one of the integrase. So integrase is a big family of enzymes. So BXB1 is highly specific. The reason we chose this is that BXB1 is highly specific integrase. And we wanted to explore the use of BXB1 in the application of gene therapy. So then, Sense Foundation teamed up with us, and so we made this maximally modified mouse named by Aubrey, 3M mice, and these animals can now be used to test for gene insertion. So basically, these animals contain BXB1 recognition site into the mouse genome. So we inserted it into a couple of safe harbor locus in the genome, and then now we can test any gene of interest to insert at a specific location. So for example, if the gene is a green fluorescence protein, and you will see green mice, and if it expresses. So this is the project that we're working with Sense, and currently we're testing the insertion of various fragments into the mice. So, and then there are many things that we need to do, for future directions. So, just raising a few of them. For example, how to deliver our integrase into the human cells, and eventually we need to do more protein evolution to direct BXB1 to human genomic sites, and they may be off target, so that's for safety reasons we need to understand, and there are many others. So I'm going to skip all this, and if you're interested in knowing more what we do, I will be very happy to talk with you. So here I'm stealing a slide from George Church. So he actually gave a talk in an Integrative Medicine Network forum. And so they have discovered 45 age-reverse genes. So that's his proposal, is to introduce all these anti-aging age-reverse genes at once. And using AAV, that's what he proposed. And to potentially to reverse aging. So that's a very challenging task, but people like him is already thinking about doing it. All right. So the last slide. So most work is done at Applied StemCell. We are located in the sunny San Francisco Bay Area, and we have about 85 employees. I'd also like to thank Sense Foundation for funding for making the animals, and we also receive NIH grants. My collaborators at Stanford, Dr. Luo, Michelle Kalos, and Mark Kay. And thank you very much for your attention.
Thank you, Professor. We've got the trusty mics up there, and maybe some of the material that you did get.
So I have a question. I mean, in comparison to kind of wild-type BXB1 and Phi integrases, what enhancements Target integrases have?
The BXB1?
Yeah, I mean, wild types that, for instance, I can get in gene, or that was published in Kellas dice plasmids. Did you make any direct, I mean, those that you sell right now, is it wild type or they are enhanced?
Right now, we're still at a wild type, but actually we're evolving the protein. Now we're going to actually move to enhanced. So the enhanced protein, the enzyme has to recognize the natural sequence in the human genome so that we can use directly into human therapy. So that's a long process. We've been doing that, the direct evolution, for over 3 years. And we have intermediates now that works in a human sequence.
Thanks for the talk. Thank you very much. So I have a question about the price of the therapies that you develop. Should we expect a drop in price anytime soon in the near future?
Yes, that has to happen. So basically, the major bottleneck for gene and cell therapy is manufacturing, so GMP manufacturing. So that process needs to be streamlined and the cost needs to be lowered. Otherwise, I think it's going to be very difficult to, especially in the US right now, insurance pays for it. There are different ways of doing it, but in other countries, then there's no way. In developing countries, people cannot afford it.
Thanks.
And last question here.
So in mice, there are very well-established safe harbor regions, but where are you going to integrate in human cells?
Very good question. So we have identified, we actually published a paper on that, safe harbor loci in a mouse. Then we looked orthologous sequences in humans. So we have identified several in a human that also works as a safe harbor locus.
So you already checked that they give stable expression and don't change expression of it?
Yeah, we published that already in iPS cells and ES cells.
Okay, thank you.
Thank you very much, Professor Chen-Tsai.
Okay.
Thank you.
Thank you.